Okay, let's dive right in. We're going to talk about how a truly revolutionary new technology is starting to crack one of the oldest, toughest, and most important puzzles in all of biology. So here's our game plan. We'll start by understanding the biological puzzle itself. Then, we'll look at a brand new quantum tool built to solve it. We'll see how scientists had to fight through something called quantum noise. Then, we'll see them put it all to the test on a real protein. And finally, we'll get a glimpse of what the future of folding might look like.
So first things first, let's really get our heads around this problem that has been a brick wall for scientists for decades.
And this isn't just some academic question, right? The answer has massive implications for our ability to design new drugs, new medicines, and to really understand the tiny machines that make life itself possible.
The puzzle is protein folding. You see, when a protein is first made, it's basically just a long, floppy, and pretty useless string of amino acids. It's only when it twists and contorts itself into a very specific, very intricate 3D shape that it can actually do its job, whether that's fighting off a virus or making a chemical reaction happen inside our cells. So what makes this so mind-bogglingly hard? Well, it's the sheer number of possibilities. The number of ways a single protein could theoretically fold is, well, it's astronomical. For a pretty average-sized protein, there are more possible configurations than there are atoms in the entire known universe. In computer science, they have a name for this kind of problem, NP-complete, and that's just a fancy way of saying it's a total computational nightmare.
For years, we've been chipping away at this with our classical tools. Lab experiments, which are incredibly slow and expensive. And even our most powerful supercomputers, running simulations, can take months just to model one protein. The reality is, our classical tools just aren't built for a problem of this scale. We need something totally new. And that, right there, is where quantum computing steps onto the stage. So what does this new quantum toolkit actually look like? Well, at its very heart is a really clever algorithm that researchers are getting pretty excited about. It's called the Variational Quantum Eigensolver. I know, it's a mouthful, so we'll just call it VQE. The best way to think about it is as a smart partnership. The quantum computer does what it does best, the really heavy lifting, exploring a huge number of possible folds all at once. Then it hands its results over to a regular classical computer, which analyzes them and says, okay, that's interesting. Now try looking over here. It guides the quantum search, pushing it toward the most stable, lowest energy shape, which is the one we're looking for. But does this hybrid approach actually work any better? Well, the proof is in the pudding. A recent study put it to the test on a set of 50 small proteins. The traditional simulations, they found a stable fold about 72% of the time. Not bad. But the quantum VQE approach, it succeeded a staggering 98% of the time. That's not just a little better, that is a huge leap forward.
Okay, as amazing as that sounds, there's a catch, and it's a big one. Today's quantum computers are, well, they're noisy. They're incredibly sensitive. Tiny vibrations, tiny temperature changes, they can all create errors in the calculation. It's kind of like having static on an old phone line that just scrambles the message. And that can lead you to a completely wrong answer. This challenge forced the researchers to get really, really creative. To solve the problem in biology, they realized they first had to solve a problem in quantum physics. They had to figure out how to build a better, more noise-resistant quantum circuit. So they set up a head-to-head comparison. The standard circuit design relies on a set of basic quantum operations called Clifford gates. But their new, innovative design added a special ingredient into the mix, a much more powerful operation called a T-gate. Now, T-gates are known to be absolutely essential for building the fault-tolerant quantum computers of the future. And the result of adding that one little component was, well, it was incredible. With the standard circuit, the error rate was almost 7%, and it took about 70 attempts for the algorithm to finally settle on an answer. But by just adding that one T-gate, the error rate plummeted to just 1.4%, and it found the answer more than 10 times faster. To really nail this point home, check this out. That single change, just adding the T-gate, was responsible for almost 90% of the entire performance improvement. This wasn't some minor tweak. This was a massive, massive leap forward in the fight against that pesky quantum noise. All right, so now they've got this souped-up, noise-resistant quantum tool. It's time for the main event. They decided to put it to a real-world test, folding a part of a really important human protein called ubiquitin C. And they didn't pick an easy target either. They specifically focused on the tail end of this protein. This part is famously intrinsically disordered, which basically means it's a wiggly, flexible bit that doesn't have one single fixed shape. That makes it incredibly difficult to model, but that flexibility is also what makes it so important for how ubiquitin C works in our bodies. So they ran the experiment on 2 systems. First, on a simulator that was deliberately noisy, and second, on a real quantum processor, the Aria-1, which has error mitigation built right in. And just look at the difference. The real, error-corrected machine spit out far fewer, much cleaner results. It narrowed the possibilities down from 145 different potential shapes to just 53. It found the signal in the noise. When they dug into the details, the problem with the noise became crystal clear. Over 5 identical runs, the noisy simulator gave them 5 completely different answers for the protein's final shape. I mean, imagine asking a calculator, what's 2 plus 2, 5 times, and getting 5 different answers. That's, well, is totally unreliable. But then you look at the real, error-mitigated Aria-1 quantum processor, and this is the magic moment. Across 5 identical runs, it produced the exact same consistent fold every single time. That right there is the power of that T-gate innovation and fault-tolerant design in action. It's consistency, it's reliability. So what's the bottom line here? What's the big payoff? Speed. The quantum method found a stable, reliable fold in just under 11 minutes. A classical supercomputer simulation took 6.5 hours to get to a similar point. Now, to be fair, the classical simulation did eventually find a slightly more stable structure. But think about that trade-off. The quantum computer delivered a reliable, consistent answer in a tiny fraction of the time. That is a game changer. This is a huge proof of concept, a real landmark. So, where do we go from here? What does the future actually hold, and what hurdles are still in our way?
Well, let's be real, we're not about to cure Alzheimer's tomorrow. Today's quantum computers just don't have enough stable quantum bits, or qubits, to handle really big proteins. For example, even a small molecule like aspirin would need about 146 qubits, which is more than we have right now. Plus, as these systems get bigger, the algorithms themselves can run into problems with accuracy, a strange phenomenon that scientists call the barren plateau. So the path forward is actually pretty clear. It's going to require a 3-pronged attack. First, designing even smarter hybrid algorithms, maybe combining the best parts of different quantum approaches. Second, the obvious one, continuing to build bigger, better, and more stable quantum hardware. And third, integrating all of this quantum power with the incredible pattern recognition abilities of AI and machine learning. So even though real challenges definitely remain, this research shows us a clear path forward. It proves that quantum computers are evolving from being these theoretical physics lab curiosities into becoming genuinely powerful, practical tools for scientific discovery. And it leaves us with a pretty inspiring question to ponder. Once we finally do crack the protein folding code, what disease do you think we could cure first?